Hello everyone, I'm Dr. Amna Khan. I have completed my master's in oral maxillofacial surgery. Now let us discuss something about the process of digestion. The process of digestion is accomplished by mechanical and chemical process. The buccal cavity performs two major functions, that is the mastication of food and facilitation of swallowing. Now the teeth and the tongue, with the help of saliva, masticates and mix up the food thoroughly. The mucus in the saliva helps to lubricate and adhere to the masticated food particle and form a bolus. Now this bolus is conveyed into the pharynx, which is further pushed down to the esophagus by the process of swallowing or deglutition. The bolus further passes down to the esophagus by successive waves of muscular contraction called as peristalsis. The saliva in the oral cavity consists of electrolytes and enzymes, the salivary amylase and the lysozymes. The chemical process of digestion is initiated in the oral cavity by hydrolytic action of the carbohydrate splitting enzyme, the salivary amylase. About 30% of the starch is hydrolyzed here by this enzyme in the optimum pH of 6.8 into disaccharide, that is maltose. The lysozymes present in the saliva act as an antibacterial agent that prevents infection. Now, the mucosa of the stomach has gastric glands. The gastric glands have three major types of cells, namely the mucous neck cell, which secretes mucus, the peptic or the chief cells, which secretes the proenzyme or the pepsinogen, and the parietal or oxyntic cell, which secretes the acyl and the intrinsic factors. Now, these factors are essential for the absorption of vitamin B12. The stomach stores the food for 4 to 5 hours. Yet the food mixes thoroughly with the acidic gastric juices by churning movement of the muscular wall and is called as chyme. The proenzyme pepsinogen, on exposure to hydrochloric acid gets converted into active enzyme pepsin. Now this pepsin converts the protein into proteases and peptone or the peptides. The mucus and the bicarbonate present in the gastric juices plays an important role in lubrication and protection of the mucosal epithelium from the excoriation or by the highly concentrated hydrochloric acid. The hydrochloric acid provides the acidic pH of 1.8. Renin is a proteolytic enzyme found in the gastric juice of the infant, which helps in digestion of the milk protein. Now, various types of movements are generated by the muscularis layer of the small intestine. These movement helps in thorough mixing up of the food with various secretions of the intestine and thereby facilitates digestion. The bile, pancreatic juice and the intestinal juice are the secretion released into the small intestine. Now, the pancreatic juice and the bile are released to the hepatopancreatic duct. The pancreatic juice contains the following enzyme: the trypsinogen, the chymotrypsinogen, carboxypeptidase, the amylase, the lipase and nucleases. Now the trypsinogen is activated by an enzyme called as enterokinase into active trypsin, which further activates other enzymes in the pancreatic juice. Now, the same way, the bile contains the bilirubin or the biliverdin, the bile salt, cholesterol, and the phospholipid. These helps in emulsification of fat and also activates the lipase. Now, the intestinal mucosal epithelium has goblet cell which secretes mucus. The secretion of the brush border cells of the mucosa along with the secretion of the goblet cells constitute the intestinal juice or the succus entericus. This juice contains a variety of enzymes such as disaccharides, example maltase, dipeptidase, lipases, and nucleosidases, etc. The mucus along with the bicarbonates protects the intestinal mucosa. We have discussed this earlier, and provides an alkaline medium, that is pH of 7.8, for enzymatic activities. The submucosal gland or the Werner's gland also helps in the enzymatic activities. The protein, the proteoses and the peptones, these are the partially hydrolyzed protein in the chyme, on reaching the intestine are acted upon by the proteolytic enzymes of the pancreatic juice, such that the protein, peptone and proteoses in the presence of trypsin or chymotrypsin, it converted into dipeptides. The same way the carbohydrates in the chyme are hydrolyzed by the pancreatic amylase and is converted into disaccharides. Fats are broken down by the lipases with the help of the bile into di and monoglycerides. The nucleases in the pancreatic juice are acted upon on the nucleic acid to convert it into nucleotides and nucleosides. Now, these enzymes in the succus entericus act on the end product of the above reaction to form the respective simple absorbable forms. These final steps in the digestion occur very close to the mucosal epithelial cells of the intestine. Now, these biomacromolecules, as it mentioned here, is these breakdown happens in the duodenum region of the small intestine. The dipeptides form in presence of dipeptidase forms amino acid. The maltose in presence of maltase is converted into glucose and glucose. The lactose is converted to glucose and galactose. Sucrose in presence of sucrase is converted to glucose and fructose. The nucleotides in presence of nucleotidase is converted into nucleosides, which further converts to sugar and bases. The di and monoglyceride which were formed in presence of lipases is converted into fatty acids and glycerol. Now, the simple substance thus formed are absorbed in the duodenum and ileum regions of the small intestine. The undigested and unabsorbed substance are passed on to the large intestine. Now, what is the function of the large intestine in the digestion? Firstly, absorption of some water, mineral and certain drugs. Secondly, the secretion of mucus, which helps in adhering the water, waste, undigested particles together and lubricating it for an easy passage. The undigested, unabsorbed substance called feces enters into the cecum from the large intestine through ileocecal valve, which prevents the backflow of the fecal matter. It is temporarily stored in the rectum till defecation. Now, the activity of the gastrointestinal tract are under neural and hormonal control for proper coordination of different parts, like the sight of smell or the presence of food in the oral cavity can stimulate the secretion of saliva. The gastric and intestinal secretion are also similarly stimulated by neural signals. The muscular activity of different parts of the alimentary canal can also be moderated by neural mechanism, both local and through the central nervous system. The hormonal control of the secretion of the digestive juices is carried out by the local hormones produced by the gastric and intestinal mucosa. Thank you.